Hi, this is Mrs. Nagayama, and I'm going to go over quiz 2.19 today. Please review sections 2.17, 2.18, and the review on 2.19 before taking this quiz. So I just wanted to review very quickly monomers versus polymers. So monomers, remember, is the smaller subunit of a polymer. So if I said that a monomer was a brick, then the polymer would be something like a brick house or a brick wall. Again, a monomer is a smaller subunit or component of a polymer. So if we're thinking of a different type of macromolecule, like proteins, we obviously will have a new monomer. So the monomer for proteins are amino acids. So if you see on the right-hand side, you have a red circle, and that represents one amino acid, which is that small subunit that makes up a protein. If we were to string all of these red circles together, or red amino acids together, then what we would form is a protein. So a long strand of amino acids make up a protein. The average number of amino acids in a protein is about 500, but a protein can have as many as 35,000 amino acids. So again, the small subunit for a protein is amino acid. When you link all those amino acids together, we now have our polymer, which we call the protein. So a sample question for quiz 2.19 is, which repeating units make up a protein? Another way to say repeating units would be subunit or monomer. So what is the monomer of a protein? We see that in the slide just before that the monomer for a protein, or each red circle, represents an amino acid. The answer for this question would be C, amino acids. All right, so if we look a little bit more carefully at the amino acid structure, we'll see that they all have the same amino group, which is highlighted in blue on the left, the same carboxyl group, which is highlighted in green, and the hydrogen, which is highlighted in purple. So all of the amino acids will have the same amino group, carboxyl group, and hydrogen. However, amino acids have different R groups. So R groups are highlighted in the left picture in yellow. So the R group is what we call the variant because it's different from amino acid to amino acid. So if you look on the right-hand side, we have two examples of amino acids. We have the glycine, and we also have alanine. So both of these, if you see at the top, it says that it has COO negative. That's what we call the carboxyl group. They both have that. On the left-hand side of both of them, they have an H3N, and that is the amino group. They both have that. And on the right-hand side, they both have a hydrogen. Just like we said before, all amino acids have those three things: the amino group, the carboxyl group, and the hydrogen. However, if you see highlighted in red, with a red box around it, we have different R groups. So we have a hydrogen for glycine and a CH3, or a methyl group, for alanine. So we see that amino acids have the same structure except for their R group. So we talked about how amino acids are monomers, right, which means that they are repeating subunits, right, that are going to make up a polymer, which would be our protein. So in order for these amino acids to actually stick to one another, they have to form peptide bonds. So peptide bonds hold amino acids together. So you see we have 3 amino acids on the left-hand side, and in between each amino acid is a peptide bond that's linking those two together. All right, now we're going to go into examples of proteins. So one example of proteins is enzymes. Enzymes are proteins that speed up reactions. So to kind of give you a glimpse of how this happens, if you think about making DNA in your body, that's one of the most important things that your body needs to do. Without an enzyme, it would take you 78 million years to make DNA in your body, which basically means life would not be possible. However, with enzymes, making the DNA in your body can happen in less than a day. So you can see that enzymes speed up reactions not just by 2 times or 10 times, but is a difference between 78 million years and 12 hours. So enzymes are obviously very important for life. But how do enzymes speed up reactions? So what I want to show you guys here is this graph. So you see that we have a red line and we also have a blue line. The red line is a bigger hill than the blue line. So I want you to imagine that you're pushing up a wagon or pulling up a wagon up a huge hill. It's obviously going to take a lot more energy on the red hill than on the blue hill. And so what enzymes do actually is they lower that hill. They make it a smaller hill so that the reaction can happen faster. The reaction cannot happen until it gets to the top of the hill. So you can imagine that on the blue line, it's going to happen a lot faster. What we call that hill in scientific terms is activation energy. Activation energy is the energy that it takes to start a reaction. So what enzymes do is they lower that activation energy or lower that hill so we can do the reaction a lot faster. So enzymes speed up reactions by lowering activation energy. So here's a sample question from quiz 2.19. Which sentence best describes how enzymes speed up a chemical reaction? Enzymes are not is it A, enzymes are not changed as a result of a chemical reaction. Enzymes add heat to a chemical reaction. Enzymes remove heat from a chemical reaction, or enzymes lower the activation energy of a chemical reaction. Look at the slide that I just showed you. How do enzymes speed up reactions? They lower the activation energy. Answer for this question would be D, enzymes lower the activation energy of a chemical reaction. All right, and then the last concept that we're going to go over is that enzymes only work in certain conditions. So enzymes are very important and they do make reactions happen a lot quicker than they would if the enzymes didn't exist, but they also need perfect conditions to work. So they need to be in a certain temperature, and they also need to be in a certain pH. So it can't be too acidic, it can't be too basic. It has to be very specific to the environment that the enzyme likes to work in. So if the conditions for the enzymes are not perfect, then it will not speed up reactions efficiently. So if, for example, an enzyme loves to be in an acidic environment, but the environment turns into a basic environment, then what will happen is the enzyme will shut down and not work as efficiently as it should. So knowing that, here's the last question. The human stomach contains acids that aid in digestion. A stomach enzyme was placed in a solution that contained no acid. Why did the stomach enzyme not work perfectly? So we have an enzyme that usually is an acid, but then we place this enzyme in a solution that contained no acid. Why did the enzyme not work properly? Is it A, because it requires certain conditions to function effectively? B, it cannot work outside of living things? C, it needs an energy source? Or D, it breaks apart after they speed up a reaction? So if we revisit the slide, we'll see that if conditions for an enzyme are not perfect, then it will not speed up reactions efficiently. We saw that the enzyme usually is an acidic environment, but the stomach enzyme was placed in a solution that contained no acid, so we're changing its environment. So the best answer for this one would be A, enzymes require certain conditions to function effectively, and that's why the enzyme did not work properly. All right, so that concludes the review for quiz 2.19. Remember to review the sections that I went over in the beginning before taking the quiz. And even if you're not happy with your score, remember you can always retake it. Your goal is to always get a 60% or above on all your tests and quizzes because that's a passing grade in the class. Please email me or contact me if you have any further questions.